Good evening, everyone. We are ready to begin the third guest lecture of the annual lecture series organized by the Chemical Society of University of Peradeniya. As you already have been informed, today's lecture is based on the topic Smart Materials for Molecular Recognition, conducted by our guest speaker, Assistant Professor Rohan W. Gunasekara, Department of Neurology, Yale School of Medicine, Yale University, New Haven, Connecticut, USA. Let us directly move on to the procedures. It is my pleasure to welcome all of you to this fascinating lecture, which I am sure every one of you is going to find exciting and beneficial. First and foremost, I warmly welcome our guest speaker, Assistant Professor Rohan W. Gunasekara. Next, I would like to welcome Head of the Department of Chemistry, Professor Manadevi Ganehenege, and the Senior Treasurer of the Chemical Society, Professor Dr. Tiloka Aryasena, whose guidance and support has been an inspiration for us to organize this event. In addition, I would like to welcome the lecturers from the Department of Chemistry and the lecturers from the other departments, demonstrators, and all the students who have joined us to benefit from this lecture. I am confident you will find the lecture inspirational, exciting, and beneficial. Next, I would like to welcome Dr. Tiloka Aryasena, the Senior Treasurer of the Chemical Society, to introduce our guest speaker.
Very good evening to you all, and then I must say good morning to Roshan. I'm happy to introduce Dr. Rohan Gunasekara from Yale University today. Dr. Roshan graduated with a BSc Special Degree in Chemistry from University of Peradeniya in 2010. Then he obtained his PhD degree in Chemistry from Iowa State University in 2016. Thereafter, he went to Department of Neurology, Yale University, as a postdoctoral research associate in 2016, where he continued in the position from 2016 to 2018. He worked as an associate research scientist in the same place from 2018 to 2019, and he became a research scientist in the Yale University, the same department, in 2019 and continued till 2020. He's working as an assistant professor from 2020. Dr. Roshan won the Frank J. Moore and Corinne Beth Moore Fellowship Award in 2015, and he won the Graduate Research Symposium Award and the Alpha Chi Sigma Research Award in 2016. He has won the Yale Center for Clinical Investigation Awards in 2020. Impressive achievement, Dr. Roshan. Without further ado, I would like to invite Dr. Roshan to deliver his lecture. Over to you, Roshan.
Can you see my slides?
We can see, Roshan.
Yes, we can hear, Roshan.
Okay. Thank you very much, Dr. Thiloka and Pasindu, for the kind introduction. And also, I'd like to thank all the Chemical Society members and all the members of the University of Peradeniya who actually invited me for this guest speaker. And it's been my privilege to be here and delivering such a presentation. And also, before moving to content, I would like to tell you as a general principle, I have already broken down all these slides so that anybody have a background of understanding electrostatic interactions, hydrophobic interaction, and hydrophilic interaction would definitely understand my research work. So without further ado, we will go to my content. So in my content, I would like to first talk about a little pathway to a PhD and what is beyond PhD in the United States. And then I will switch on to Yale University and what I do here as an assistant professor. And then finally, then I will go to the main core that is about my prior research work that I did at Iowa State University. So moving on to a PhD and how we can do a PhD program here at USA. So you can see this slide where if you are I think I'm pretty sure you might be belong to one of these category. So 4-year degree with a BSc, actually, if you are in special degree, you are being qualified for a US PhD program. I mean, on the other hand, don't get worried if you have a 3-year degree in BSc. There is another path, compensate and jump into PhD program by doing MSc degree for 2 years. And also I didn't include another pathway that if somebody is doing MPhil, also quickly jump in to the PhD program without further ado. And the other thing is PhD program in United States, you need to have certain qualifications in GRE, TOEFL, uh, degree itself and transcript, some of and recommendation letters from the faculty. So if you have all these qualifications, definitely you will be able to do a PhD program. In United States of America, the PhD program is consists of two parts. One is the master's degree program, and then the component, which leads to at least 5 to 7 years doing a PhD here. And after finishing your PhD, you are either eligible to go to industry directly, or you have the privilege to go to a postdoc in another university. So if you go to industry, the industry has two types of sections, like called the pharmaceutical side or the biotech side. You can join the tech side, or you can join the pharmaceutical companies, and then depending on your profession, you can continue learning and then move up the ladder inside the industry. In the other way around, if you go to postdoc position, you have to do another research, maybe for another 2 to 3 years, choose to go to industry or faculty. You might have a question why we have to do, what is the difference between actually the PhD program and the postdoctoral position. The main difference between a PhD program and a postdoc in United States is that in PhD program, your professor in charge will train you with all the skills. Actually, it's going to be like a spoon feeding system. But when you come to postdoc level, you independency, so that you are the kind of the driver of your research component. So therefore you have a little bit of flexibility, and you the one who is driving your research towards the end goal. So that is the difference between PhD program and postdoc. But to accomplish a good postdoctoral position, you should have the skill sets that is directly coming from the PhD program. So after completing 2 to 5 years, depending on your research component, you can again into industry, or you can join to faculty. So here that where I'm situated, and the good thing about the faculty is that still in the faculty, you have a ladder process in United States, as you can assistant professor, and then you can promote to associate professor, and then you will get the full professorship. If you still want to go to industry, there is always a pathway to go and join industry. So this is the simple version of getting a PhD from United States, or what is beyond that you want to do after accomplishing a PhD program. So with that, I will move on to the Yale University. So this is where I'm working on. So the Yale University is a private Ivy League university that was established in 1701. So this is the third oldest university that is in United States. Of course, the most oldest university is the Harvard, and then the third one is the city. I mean, at this 2020 October, you can see there are about 65 Nobel-oriented prize winners affiliated with Yale University. According to the national ranking system, the Yale University is among the fourth. And I have put a picture of Yale University because this is the main library of the Yale University, which is having several stories building. So moving forward, Yale University is about history and education. So if you think of things and the history, it's go back to 1701. And in fact, I'm choosing here some of the pictures because I couldn't include all the pictures though. So you can see here physics, engineering, and forestry management, and also the gymnasium. So I want to talk about a little bit of the chemistry and physics departments in Yale University, because I think you guys have heard about Gibbs. So the Willard Gibbs studied thermodynamics and discovered statistical mechanics. And he discovered all these thermodynamic statistical mechanics here at Yale in chemistry and building, and they have allocated a special place to identify his recognitions. On top of that, we have a gymnasium. I want to talk about a little bit of the gymnasium as well. It is one of the largest athletic facilities ever built, 12 acres of interior space. Even though this small, it is 12 acres of interior space, including 9-story tower containing the third floor swimming pool. So this is very rare situation. I think it's one of the rarest thing that you can in Yale. Not only that, I chose to show you some of the other pictures like uh, the low building, architectural building, Bernanke School of Medicine, and Peabody Museum. So I chose this building because this is the second library, the main library in Yale, and it has some significant value. The value is that this building contains rare books library, and it also stores one of the most promising and most important articles inside the library. You can see this is made out of actually marble. With stones or anything else, it's just been made out of marble, just because protect from UV light, and this entire building is lighted up by electricity, just to protect from UV light. And the other thing is the museum. The Yale is also hosting 3 by its own expense. That is because they are having a different. And one of the important thing about this Peabody Museum is it's the national history at Yale University, and it's among the oldest, largest, most prolific university natural history museums in the world. And if you want to talk about a Peabody Museum, this is actually encapsulating one of the dinosaur skeletons, and as well as mummies inside this museum. So that is why it is been world, and it's been, I mean, it's been taken care by the Yale University. And this is where I'm working on the School of. So this is actually a big building, and that is where I conducted my research work. So moving on to my research work at Yale. So you can think of that, how can I jump into neurology from chemistry? So actually, when you learn the skill set from the PhD work, you have enormous freedom to do whatever you learn. So that is why I chose neurology after my PhD, to come and join and to learn and see how the brain works in conditions. So the brain, I mean, is like this, and it is made out of arteries, arterioles, and capillaries all over the place. The brain is complex structure. It is working uniquely with the help of these cell types. So if you take one section of this capillary, it made out of these prominent cell types. So my is to develop novel brain cell specific chemicals that can be used as PET imaging and as well as for transportation into these particular cell types. So to do that, I lot of cellular functions, brain functions during my postdoctoral research work. And not only that, I had chemical work as well. So my lab right now at Yale is a combination of chemistry and neurology. And it is a highly synergistic fashion. And we are kind of solving complex problems that is related to the neurological diseases. So one thing that I'm doing in my lab is I do a lot of synthesis. This synthesis doesn't exclude any of the prominent. It includes all of the chemical book synthesis, including organic reactions. You can imagine of the inorganic reactions. And whenever you do all these reactions, before doing any testing in vivo in cell culture, we do a purification to find out the pure material. So for the purification, we have automated two instruments, one called the Biotage and the other one called the Agilent system. And then comes the characterization part. You will learn normally in a BSc degree. So every compound can be characterized through NMR or mass. So this is actually one of the 12th one of the 12th NMR facilities that is there in Yale, and this is actually a mass. So this is actually not situated in my lab, but we use co-facility in order to get the service and also to get the job done. And then we have a separate housing for in vivo experiments. We are housing actually rodents in our lab, and we do in vivo and we do cell culture work. And also on top of it, we have full access to do with the use of either confocal microscopies or two photon microscopies. So this is combining the chemistry and neurology, the brain functions in order to understand how they function and in order to get solutions from chemical point of view. So the molecular recognition, you can think of any molecule that you can identify. So you have to develop a certain skill set in order to identify these molecules in solution. So to develop this type of identification in solution, we had two technologies brought up in our lab. One is called the cooperatively enhanced receptors. This is a receptor that works to identify aromatic amines and citric acids particularly. And then we jump into another technology called molecular imprinted nanoparticles. That is a very easy and is a diverse method that can be used to identify sugars, peptide, glycolipids, urea. But not only that, but you can think of anything. After this presentation, you will realize the molecular imprinted can be used actually for recognizing many more substituents that you can think of. Let's move on to the cooperative enhanced receptor part first, and then we will visit the nanoparticles later. So cooperatively enhanced receptors is generally being seen in nature. So one of the prominent example is the biotin streptavidin complex. You can think of this binding energy. The guest is the biotin molecule. The streptavidin is the protein that is known to be the host complex. So when they make this host guest complex, the binding cost jumps by out of 40. So that means this binding energy is enormous that when they are come together to make the complex, it is so powerful so that it requires super amount of energy to break it off. Not only that, the stability is demonstrated by the melting point. The melting point increases by 30 Celsius degree after making the host gas complex. That means after making the host gas complex, which is biotin streptavidin complex, the stability increases so that you need high energy to break apart. So the scientists were looking at how we can demonstrate this type of binding constants in chemical methods, and how we can implement this type of binding energy through organic chemical structures. So we came up in our lab as a hypothesis. The hypothesis is very simple. So you can think of in the slide, this is known to be as cooperatively enhanced receptor, the CER, that is mentioned here. Of this cooperative enhanced receptor as a human arm. So which is exactly what we wanted to demonstrate. So this one, A and A dash, is your arm, and think of this B as your fingertips. So when the incoming guest or whatever compound comes in close, what it happens is that the AA dash interacts each other. It makes a direct conjugation or direct complex between G and the host B. So that is what it happens. So think about this arm, and when it is getting close and closer, there will be A and A dash hydrophobic interaction takes place. This is called hydrophobic interaction, while there will be an direct electrostatic interaction G and B. So we want to demonstrate that the B electrostatic interactions were driven by the hydrophobic AA dash interaction. So for that, we developed a synthetic chemical method to design and synthesize this type of molecules. So let me explain about this molecule. This molecule is called the cholic acid. The blue one is beta phase. The alpha phase is the hydroxyl part. The part is super hydrophilic and it's water soluble, and the beta is hydrophobic. The beauty of this cholic acid is that it is water soluble. So, and also it is lipid soluble. So it's either soluble in water and either soluble in lipids. So this is a beautiful molecule. That's why we took it. And we connected these two cholic acids by a flexible a tether that is called the elbow. This is exactly working as your elbow, and this red marked one is your B. That is the one that is susceptible for the incoming guest. So let's think about this molecule in a sophisticated way. Believe me, this compound 1 takes about almost 13 or 14 steps to synthesize. And the beauty of this molecule is that this molecule is fluorescent. So the inner core here, this complex is fluorescent. And not only that, this has 3 arms, 3 human arms. If you can think of, when the guest comes in as an amine, aromatic amine, when you put this amine, you will definitely think of that these amines will be interacting with the carboxylic acids at the end. So these carboxylic acids at the end will be interacting with these ones. But the good idea is not only that, there is some indirect interactions that is taken place. This is the interaction that is called as indirect interaction, that is called the hydrophobic interaction, that utilize and strengthen this direct electrostatic interaction. So this is what we wanted to demonstrate by this publication. In order to this publication, we need few more organic materials to synthesize. So we went ahead to synthesize, instead of the 3 arms, 2-arm molecule. So this is a 2-arm molecule. And this 2-arm molecule is cut down in actually a 1 arm. And then we throw the molecule like the amine here. And again, this will directly involve in electrostatic interaction with the carboxylic acids. Top of it, you will see that you will appreciate there will be a big interaction between these 2 cholic acids and these 2 cholic acids. Furthermore, we wanted to demonstrate by using a control experiment. This is the control experiment we use to demonstrate that this molecule does not have hydrophobic interactions. How? When you throw this molecule into, when you throw the same amine molecule into the system, what it happens is there will be a direct interaction between this amine and this carboxylic acid, and so forth. This amine and this carboxylic acid, this amine with this carboxylic acid. But there will be no any arm effect, as I mentioned earlier. So that means there is no indirect hydrophobic action with this control compound. So this also takes a little while to synthesize, you can imagine, and I did this synthesis by myself. So we looking at this one, let's move on to a little bit of the data. So the arm effect has to be proven in order to data. So what we did was we took the compound 1 and again we synthesized the control compound called compound 4. If you think of the compound 1 and compound 4, only difference between 1 and 4 is that have rotatable arms. These are rigid. Can you, I mean, if you can see of this compound 4, there is no flexibility. They cannot do the arm rotation. So therefore, there is no arm effect. So when you throw this compound into water methanol ratio, we understood that when you are increasing the water content of the solution, the compound 1 shows an arm effect. And when the arm effect comes into play, these carboxylic acids come close to the center of the molecule and quench a little bit of the fluorescence. And that is what you see as quenching of the fluorescence, and it stabilizes at the middle point. But the compound 4 cannot quench the fluorescence here because there is no bending effect. 50% of methanol to 100% of water, there is no quenching of fluorescence. So we demonstrate that this compound 1 molecule works exactly as a human arm in water solution. Moving ahead, now we want to characterize how the binding taking place. For the binding taking place, we are introducing one simple method that is called isothermal titration calorimetry. This is exactly similar to what you do in analytical labs to understand how heat dissipation or the heat absorbance that is taking place. So this machine is very sensitive, and this machine is loaded with a lot of host, that is the host molecule, and the guest is put in the syringe, and then you allow the machine to drop one by one the guest into the solution. When the host meets the guest, either the temperature increases or the temperature decreases. When the temperature increases or decreases, this coil, this particular coil will recognize it, and then it will give us. So these spikes will represent either heat dissipation or heat absorbance, and depending on titration, you will generate a nice curve, and you will see a plateau. When you see a plateau means your host is no more there in order to any extra guest. So that is why you will see a plateau. So the beauty of this ITC methodology is that super sensitive, and it is giving you your binding constants, accurate, reliable, and very fast. Not only that, it also gives you the N value. The N value is the number of host to guest ratio, binding constant, the Ka, delta H, and the delta S, the entropy in one single experiment. So this one experiment, you can get all of these thermodynamical properties, and you can reproduce it by doing again. And then if the values are similar, then you can think of that they are good to go to a publication. So let's look at a little bit of the data, the binding data that we are thinking of. So the compound 1, as I mentioned earlier, this is a perfect molecule that really works as a human arm. So when this molecule meets the compound 5, the amine, the aromatic amine, binding constant is 138 10 to the power of 3. And when you increase the length of this amine, just like another CH2, it drops to 11 10 to the power of 3 value. And if you go further with increasing the arm by another CH2, it goes to 8 10 to the power of 3. So these 5, 6, 7 guest molecules, we demonstrate the compound 1 has a selectivity towards the compound 5 over 6. Not only that, we wanted to understand how this one act against 8 and 9. The 8 compound, it doesn't have a rigid center because it doesn't have an aromatic center, and therefore you won't see a binding, so therefore you can't get a binding value. And the other thing is that you cut off one amino acid from the aromatic system, and you can see the value drops to 23. So having in the system is 138, having 2 amines in the system, it jumps into 23. So now this one demonstrates that this has a direct electrostatic interaction between carboxylic acids and amino group. What we have to demonstrate is that there is an indirect interaction. For that, as I mentioned earlier, we wanted to use these two compounds as control, the compound 2 and compound 3. So compound 2 binding with the compound 10, you can see the binding drops significantly, 2.2 10 to the power of 3. So from compound 1 binding to 5 is 138, but when you have 2 arms, it drops to a significant of 2.2. It's almost a significant value drop. And the last thing is comparing compound 1 with compound 3. What is the difference? The arm effect, because there is always the direct interaction between carboxylic acid and the amino groups. So therefore there will be always the electrostatic direct interaction. Matters is the absence of indirect interaction in compound 3, because it doesn't have an arm effect, because there is no hydrophobic interaction between the two colic molecules as here. So when compound 3 goes to bind to the compound 5, 24 value. So that means from compound 1 to compound 3, the hydrophobic value cuts by almost this number. This difference is, we claim that this is due to the hydrophobic indirect interactions that with these colic acids. And that is about this paper. Moving to another hypothesis from this observation was developing another idea. Instead of using 2 colic acid per 1 arm, now you can use 1 colic acid per 1 arm. But the method is very simple. The simple method is when the incoming guest comes and binds through electrostatic interaction, then now instead of having AA dash like this, now this AA dash can be combined like this, so that indirect hydrophobic interaction can be connected like this, and this is direct electrostatic interaction. For that, again, for another project, we had to synthesize another molecule. This molecule has guanidine complex, and at natural state, these cannot combine and make hydrophobic interaction because you can see there is a charge at the end. Because of this charge, the molecule itself cannot make any hydrophobic interaction. But when you throw a citric acid into the solution, the concept changes. The concept would be now this citric acid directly connects with the guanidine complex, and also at the same time, there will be an indirect interaction between these hydrophobic, hydrophobic colic molecules. So this was the concept. Actually, we extended from the previous publication to another publication. So if we quickly go to the data part, this is actually the data. The data is about compound 11 connecting with the citric acid. Binding was formed as 77.910 to the power of 3. So the compound 11 is super specific to the citric acid over compound 15. And you can see the 15 binding to the compound 11 is a binding constant of 2.1 10 to the power of 3, which is a significant drop. And when from compound 11 to 12, we did a little bit of an adjustment. The adjustment was we changed a flexible center. Instead of having a rigid center, we used to have a flexible center. When you have a flexible center, the binding values are lower compared to the compound that was as 11. And now, again, we wanted to demonstrate the hydrophobic indirect interaction. So how do we do that? We introduce compound 13. The compound 13 definitely has a interaction to form through direct interaction, that is called electrostatic interaction, but the compound 13 has no hydrophobic interaction. As a result, the values drops to 16.3. So 1770.9 is the one that I mentioned that is coming as indirect hydrophobic interaction between these three cholic acid to make the binding powerful. So this was actually the claims that we were made for cooperative enhanced receptors. So the cooperative, now you have a little bit of idea what called as cooperative enhanced receptors. The cooperative enhanced receptor is that the concept is when the direct interaction takes place, the binding is strengthened by indirect hydrophobic interactions. So far, I was able to demonstrate that there is a cooperative enhanced receptors through aromatic amines, and we can also recognize citric acid in the solution. But complexity in these molecules. For every molecular recognition, you have to go through a lot of lengthy synthetic procedures. So this was very time consuming. A PhD student, it takes a long time to synthesize these molecules, and also to characterize these molecules, it takes a massive amount of time. So we wanted to simplify this idea through another technology that is called as molecularly imprinted nanoparticles. So when you make these nanoparticles, the idea is that we wanted to keep the core or the nanoparticle, but we can use to recognize different materials in different time intervals, like sugars, peptides, glycolipids, and urea derivatives. So the idea is very simple. Again, it is published in 2010 by this group, the Hout Nature Methods paper. What it does is they have cross-linkers and monomers mixed with the template. And when you mix everything together in a perfect concentration ratio, you will get a nice polymeric material with the template intact. And when you carefully remove this template out of this nanoparticle, you will have a reciprocal cavity that is awaiting to rebind and recognize this template. So this is exactly a replica of lock and key model. So what it does is you have the lock, you have the key, with the lock and the key, you polymerize a material. And at the end, what it does, you carefully remove the key out of the lock. And what you do is now you throw this system into a solution and find out whether it is susceptible for the key that you created. It is the simple as that idea. So to demonstrate this lock and key model and to show that these nanoparticles are susceptible and very much recognizing to this template molecule, we made a molecule like this through a system called 16, 17, and 18. Don't be scared of this material, because this 16 is a kind of a soap molecule. When this molecule reaches a certain level, you will understand this will make a micelle. So this is actually a micelle in solution. The hydrophilic parts will be pointing out, while the hydrophobic parts will be in the core. So this is a perfect micelle solution that is being created with the use of 17 and the template 18. So the template is the one that is used for recognizing the material. So as that, the compound 18 is your template. With the template, you use the different concentration ratios of this, and we actually, using research material, we actually have a recipe to cook in a correct ratio so that you will get a nice bulky nanoparticle like this. The problem in this nanoparticle is that this is very, very non-water soluble. For our purposes, this nanoparticle has to be water soluble. So what we did was we decorate the surface of the nanoparticle with a little water soluble molecule. So when it is decorated with water soluble molecule, now it quickly becomes a nanoparticle with having a very high water solubility. There is another problem that we had to address. The nanoparticle or the micelles are always tend to break. Micelles are always tend to break, but we don't want our micelle to break. So therefore, what we did was we increased the micelle density having a polymerized material, and then we irradiated using UV light. When it happens, this nanoparticle no longer going to be break out. It will be rigid and it will be polymerized and it will have a very rigid backbone so that you can use this nanoparticle in later methods for molecular recognition. So this is right now, you cooked the material with the template intact. So let's look at how we remove the template. So you can see here, the nanoparticle is here. And then if you think of the template, in this case, I'm using the glucose, the sugar. These sugars are being intact by the boronic acid. The boronic acid is here, that is hidden, but the boronic acid is the one that is keeping the glucose intact here. And when you do a little bit of acidic wash, this sugar, the glucose, will be going out. What it does is, when you polymerize in the material, you polymerize the material with the shape of the glucose structure. And when you use a little bit of acidic solution, the glucose will go out, and now you will have a perfect complementary cavity that is awaiting to rebind glucose. So that is the idea. So let's look at the rebinding process. So when you go there, and it will bind like that. So that is what happens. So if I show again, you have the glucose. Now this is a kind of the empty cavity with the boronic acids here. So this is the glucose that we use to create this nano pocket. Now when you put the glucose back, it goes back and then quickly connect with the nanoparticle. So that is the concept that we use for molecularly imprinted nanoparticle. The concept is very simple. This is as simple as the lock and key model, but we use and we developed this one in our lab, and we were able to publish in a good journal. And let's look at a little bit of the data. The data is very nice, because now I told before that when you create a pocket with the glucose, it should definitely binds to the glucose. So this is the molecularly imprinted nanoparticle that was created by a glucose molecule. So this 1 to 8 is the nanoparticle that was created with the glucose molecule. So this is the same nanoparticle without the glucose template here. And now what we do is we just titrate different sugars with the same nanoparticle. Surprise, we found it's not only glucose that binds to nanoparticle, but also another sugar that also binds to the nanoparticle. So let's look at why people binding to the same nanoparticle. So we drawn all these 8 sugars in the 5-membered ring manner, and then we were carefully identifying the structural features. So the glucose, if you draw like in the 5-membered ring structure, you will see that this looks like this. So the boronic acid is the one that binds these sugars into the pocket. So the boronic acid recognizes these hydroxyl groups and these groups. And if you compare this glucose molecule with the allose molecule, they share the same hydroxyl groups here and here as I shown here. Only difference is the hydroxyl group here it's upward and this one is downwards. So this nanoparticle pocket is super specific, and it can even identify these two molecules, only a subtle change of this hydroxyl group. So we did this one using this publication that this nanoparticle susceptible for only the material that is imprinted. We are going ahead to show that there is another version. If the nanoparticle can be made out of glucose, why not with another sugar molecule, right? So we took that privilege and we imprinted the nanoparticle using mannose. So mannose is another sugar molecule. But when you create the nanoparticle with the mannose, then people should know that the pocket is susceptible for the mannose, obviously. But surprise, as before, we found this pocket is not only susceptible for mannose, but also for altrose. Again, the idea here is mannose and altrose share the same hydroxyl configuration that is used for boronic acid detection. The only difference is this hydroxyl group is upwards and this hydroxyl group is downwards. None of the other sugars been getting into the nano pocket. So that was the idea of this paper, that we showed that if you create a pocket with a particular molecule, this molecule is super specific pocket, and that pocket is only occupied by certain set of molecules, not by all of the molecules. And to further demonstrate the hydrophobicity of a molecule, we imprinted this nanoparticle with the use of this compound called compound 20. And you can see this compound 20 has a little bit of a hydrophobic part that we added an aromatic nitro group. So when we add aromatic nitro group into the sugar molecule, now the binding jumps from 110 to the power of 3 to 26.3. So if you think of as 110 to the power of 3 value, now it jumps from 1 to 26. So it's 10-fold increment because of the hydrophobicity. And you can think of that the other molecules are also now just going into the pocket, but the selectivity is very, very high, because you can see these two has low affinity towards the pocket compared to the one that is created. The MIMP that is created with 20 is attractive towards the 20 with a value of 26.3. So the beauty of this method is now you can think of this nanoparticle now no longer going to be only for sugars. This can be rapidly expand to any molecule that you think of, and you don't need much synthetic ability or a lot of energy to synthesize materials, as I mentioned in the first part of my session. So we went ahead for another publication using dimers and trimers of sugars. So these are oligo sugars. You can see the two sugars as maltose, cellibiose, gentibiose, maltulose, lactose, and maltotriose. So we created nanoparticles with these type of sugar pockets, and then we wanted to demonstrate that these pockets are susceptible to bind to these type of sugars. And as a result, we created a pocket. Here I'm demonstrating that we created a nanoparticle pocket with the use of maltose, and then we rebound with the maltose with the use of 23 maltose, and the value was 20.5 10 to the power of 3. The value was very high, and compared to others, the maltose pocket is selectively identifying the maltose itself with a value of 20.5. And you would ask question, hey, there is another molecule or other sugars that are going into the pocket. Yes, of course, because now things have changed. Now the pocket is the pocket length is previously when we were making a pocket with one sugar, the pocket was very small. The pocket here, if I go back and show, the pocket here was very small with one glucose molecule. But what we are trying to do is that now we are trying to make the pocket bigger and bigger with particular molecules, so that now there are some certain other molecules just also goes in, just like here. But again, we created pockets, as I mentioned earlier, the compound, the nanoparticle created with 84 maltose. We can rebind the maltose with the value of 20.5, as I mentioned earlier. Not only that, we create a pocket with this molecule, and then we bind again with that, and we found a specific binding to 85 with a value of 32.9, so and so forth. We create specific pockets with respect to each and every sugar here, and then we rebind everything, and we found specific certain values that were reported in this publication. The most important part is not that, and how we can use this one for applicability. So the applicability, we use this one as to demonstrate the application from this type of nanoparticles. The idea here is that we took the sugar A, B, and H. You can think of the blood groups that you have A, B, O. So similarly, the sugars are coming as A, B, and H, and A and B has 3 sugars, while the H sugar has 2 sugars. So what we did was we created nano pockets with the use of H specifically, B specifically, and A specifically. That is called as MINP-H, MINP-A, and MIMP-B. So MIMP-H is the pocket created specifically with this one, and MIMP-A is the pocket that is created with this one, and the MIMP-B is pocket that is created with B. And then we wanted to cross-check and see how the specificity lies around. So we created with a 2 sugar pocket, and you can, it's not a surprise, right? Because you have a 2 sugar pocket, and we are trying to demonstrate and try to push these sugars into the pocket, and it never accepts these sugars anymore. So therefore, when you create a pocket with H, it never rebinds with A or B because these are bigger than the sugar H. And the concept changes when you create a pocket either with B or A. Let's look at the A. MIMP-A created a pocket with A is binding at a value of 76.7, almost roughly 77. And then if you use the same pocket to bind the B, non-specific binding, it's almost half. And if you bind the H, it will be over 9.9. So this pocket recognize, whatever the pocket is, recognize specifically the sugar that you've been created. So now, as a summary, I wanted to demonstrate that this never ends here, because while I was doing my PhD, we actually, this was a kind of a nice discovery that we made. And now, even now, my current PI, the previous PI, is still making nanoparticles and making for different substituents, as well as he's also making these nanoparticles for site-specific catalysis, because these pockets are very unique for one particular molecule, and you can grab it and hold it, and then you can use this nanoparticle for a site-specific catalysis. So with that, I would like to summarize what I did so far. So the citric acid can be specifically recognize this cooperative enhanced receptor. The aromatic amines can be significantly and specifically recognize this one. The only problem between cooperative enhanced receptor 1 and 2 is that it involves heavy synthetic background. So it takes a lot of time and effort to synthesize this and demonstrate these concepts. But when you quickly jump into nanoparticle region, things changes, because now you can polymerize these nanoparticles with anything that you think of, and then you can use the same material to show the specificity. So with that, I would move on to the last slide to acknowledge my current supervisor, Jaime Grutzländer, who hired me as a, and he's continuously supporting me for my research work that I do at Yale. And all of these people actually I interact with, and they are the ones actually helping me out and also working with me to accomplish my research goals. Plus, Yan Zhao was my previous PI. He's my current PI, and this one is he's my previous PI, actually, who trained me to synthesize organic materials. So, and also before winding up, I have to these funding agencies, the YCCI, the Yale Clinical Investigation, and also the National Institute of Health at the United States of America, that is funding my projects to carry out my research. And besides all, I would like to thank you all for listening my research presentation, and I am happy to take any question that related to this presentation or any unrelated question as well. Thank you very much.